Thank you for inviting me and thank you for introducing me. My name is Axel Schmidt. I'm from the University of Bonn and I'm presenting on behalf of the decoy host genetics group. And first I would like to start with a model. So what happens if a population meets a novel pathogen like SARS-CoV-2? Then some people will get in contact with the pathogen, others don't. And host genetics will only be relevant for the ones that get in contact with the pathogen. And those that got in contact can get infected or not. And this is the first phenotype we can observe in infectious diseases, so the susceptibility. And there are examples where human genetics already plays a role. For example, in HIV, if you have a homozygous variant in CCR5, delta 32, then you are not susceptible to HIV infections. Similarly, in norovirus infections, if you have a homozygous nonsense variant in FOR2, you won't be very susceptible to norovirus. So, but if you're infected, if you'll be infected, then you could have varying degrees of severity. And this is the second phenotype we can observe. So you can have severe disease, and for example, host genetics in HIV, in the HLA-B gene can affect the disease course towards AIDS in HIV. Just to name another example for host genetics here. So in our study, we focus on COVID-19. I don't think that I need to introduce COVID-19, as COVID-19 is very familiar to you. We focus on the severity phenotype. And risk factors for severe COVID-19 are advanced age. You can see data from 2020 from the early pandemic on the right. And male sex, comorbidities such as overweight, diabetes. But also here, we can see that the twin study found a heritability of about 31%. So heritable factors seem to play a role. And what has already been identified? There are common variants that affect the severity phenotype, but also the susceptibility phenotype. And we can really divide the common variants into these two phenotypes. And if you just have a look at this double Manhattan plot, you can see this chromosome 3 locus that was identified very early that is can predispose to severe COVID-19, for example. But besides these common variants, there are also monogenic variants. And first of all, TLR7 is the most important gene. And this was first described by Alexander Heuschen's group in two brother pairs that were young and severely affected by COVID-19. And this finding was replicated in several studies afterwards. As you can see on the right, just an example. So this is really well established, so you could even test for this in clinics, I think. Okay, so for the introduction. So I said that I'm presenting on behalf of the DECOY consortium. So DECOY is the German COVID-19 Omics initiative, and this was founded in March 2020, and the aim of it is to study COVID-19 by next generation sequencing omics technologies. And that's not just host genetics, what I am presenting, but also sequencing of viral genomes, characterizing the immune response of COVID-19 individuals, patients, and their host genetics. If you would like to find more information, you can visit the website decoy.eu. And what is the goal of decoy host genetics? So we want to characterize COVID-19 by whole genome sequencing, and we got funding by the DFG and by Illumina. And we sequence in the red noted sequencing centers, Cologne, Bonn, and Tübingen. And our study is also embedded in international initiatives like the COVID-19 Host Genetics Initiative. In this project, the problem was not the funding, the problem was sample collection. We already collected samples in 16 sites, as can be seen as blue dots on the map, but there were problems because the decentralized structure of the German healthcare system and the resulting diverse ethical votes that it sometimes not cover genetics appropriately, and also the relatively low number of severely affected individuals in Germany compared to other countries in Europe. So yeah, we actually do data freezes because, as you know, whole genome sequencing data are really big data, and the data freeze 2 is the best characterized one, and there we had, for now, and there we had 951 individuals with whole genome sequencing data and phenotype data. Then after quality control, this decreased to 863, and then we assigned a population based on principal component analysis, and the biggest subpopulation that we had were Europeans with 752. And here you can see that we have 56% male, a little bit more male individuals. And the age distribution looks like this. So it's a histogram centered around about 55 years. And if we look into the phenotypes that we have, so we classified into 3, mildly affected, hospitalized, and mechanical ventilation. You can see that the trend is the more severe the phenotype gets, the older the individuals are. So what is our analysis scheme? So first we called single nucleotide variants and insertions deletions. And what we first did is association analysis of common variants, and we first focused on the exon because of computational reasons. And what was the result? So if we look at a hospitalized phenotype, we can see this peak at chromosome 3. This is the chromosome 3 locus I indicated in the introduction, and this can be replicated, but there's no other really novel finding. So this is, but this is confirming that it works, the study design works. And after this, we also looked for association of rare variants. And first of all, we looked into TLR7 because this is the best described gene, and there we didn't find interestingly, any variant that is deleterious in our cohort. And then we continued with gene-based collapsing analysis, where we considered all genes, and used the gene-based collapsing analysis as implemented in the software Regeni and Firth logistic regression. And what you actually do is you filter your rare variants based on allele frequency threshold, so they have to be rarer than your threshold. The threshold is like indicated in these 2 tables, and they have to fulfill certain functional criteria, like also indicated in the table. And then you look if an individual has one of these variants in heterozygous state or homozygous state in such a gene, and do the analysis. But there we also had power issues, not enough power, so none of the genes with the lowest p-values reached our Bonferroni corrected significance threshold in the hospitalized and mechanical ventilation phenotype. Okay, then there are several other analyses we want to further conduct. We want to call structural variants, but they are not trying to call at least right now. And we also do analysis of runs of homozygosity, but is also not finished yet. So that's it. To conclude, what I didn't say actually, in worldwide there are not many whole genome sequencing data sets on COVID-19. So this is something that makes our data set very valuable. What we did was we analyzed common and rare variants in these 752 European individuals, but due to the relatively low power, we could not find any statistically significant association. We are currently still increasing the sample size, and we still have sequencing capacity. So if you were severely affected, or if you know of samples, please get in touch. For example, the Bosco study would be an example. And if we are finished, we will deposit our data in the Human Phenome Archive. Yeah, this is the plan. So thanks for your attention, and I would like to thank the Decoy Host Genetics group that weekly meets to discuss the data, our funders, and all the contributors. Yeah.
Thanks for your interesting talk. Are there any questions from the audience? Yes, please.
So, first of all, thank you for the interesting talk. So, I'm just looking Ringsburg.
Could you use the microphone, please?
Yeah. Sure. Perfect. So, thank you for your interesting talk. I have one question regarding, because you said that you did not find a genome-wide significant result. Was that in the end something where you say, okay, that is expected because you don't have enough power, or is it, did you do a power calculation beforehand that you say, okay, we need an odds ratio of that, and then we have enough samples? Or is it that you just say, okay, with now 700 samples, we cannot identify anything anyways, or?
Yeah, so to be honest, we haven't done a power calculation because at the beginning we really didn't know what we were looking for, or what we should look for. And yeah, we hope that with increasing sample size we could find something. But I also said that we also contribute to international collaborations, and there are other exome sequencing cohorts that also do the same analysis, and then we do meta-analysis, and this hopefully will be fine to get the power. In this meta-analysis, they then identified TLR7. Yeah. So
okay, thanks.
There's another question.
So Nick, thanks a lot for this nice overview. So I have a question on TLR7, obviously. So while I fully appreciate and may not even expect that you find a significant enrichment for coding variants, do you have any rare variants in or around the gene in man? Because I would kindly offer to test that functionally. Even a singleton hit could potentially be very interesting.
Okay.
So while you never reach significance in a statistical point of view, I think the functional assays are quite robust by now, and we can test that. And I would dare to say that there must be structure variants in and around the gene or point mutation somewhere, and it would be worth, and the test is relatively straightforward.
Yeah. So I plan to really cut out the regions of the PAM files and really analyze it in depth because I also can't believe it that there's nothing in this cohort. So if there's something, we'll get back to you. So especially
fantastic. And especially in young men. So the current estimate would be that even severe loss of function should be present up to 5% of all men with severe COVID-19. If you look at men under 50 years of age.
Yeah.
Thanks.
Further questions? May I ask? Oh, go ahead, please.
Do you differentiate between the different types of SARS-CoV-2 that the patients were infected with?
Currently not, because most of the samples were, I do not have a slide for this, but most were recruited, I think early in the pandemic at least, but we have to do this in the future, if we sequence more,
Okay. Thank you very much for your talk.